Okay, we ended last lesson on speaking of measures. So we come back to them. We were going through this table, which displays several elements, several features of measures and measurements that we're going through and we'll discuss one by one. So we are talking about accuracy, and I remind you, we have said that accuracy is not synonymous with precision. In common talk, precision and accuracy are just about the same, but when we talk about measurements, they are entirely different and independent, in fact, properties of measures and measurements. Accuracy, as we said last time, refers to the vicinity of the closeness of the measures to the true value. We said that the true value does not really exist, but it's the best values that we can determine with the best available methods. So, we'll talk about what is the best practice to determine accuracy, but first let's talk about precision. Precision is the repeatability in replicate series of measurements and reproducibility on different days and in different labs. Okay, so we can depict accuracy and precision perhaps in this way. Let's assume that the true value we're trying to measure, or we are shooting at, is the center of the target. And so our shots come fairly close to the center, sometimes closer, sometimes less close, but altogether they get to the target, and their average lies just about within the true value. So we can say that to some extent these measures are accurate because their average is the true value, or is very close to the true value. In this other target here, the dispersion of the various measures is just about the same as this one. So the average distance between the measures is essentially the same. But you see obviously that their average lies about here and not at the true value. So these two sets of measures have the same precision, the same repeatability, reproducibility, which is expressed by the distance among them and between each one of them and their average. However, this set of measures is also accurate, again, to a certain degree, but this is not, because obviously the average is far away from the true value. Now, this already should tell you that accuracy should show you that accuracy and precision are not the same thing, and in fact can be separated. So let's do a little exercise here. Somewhere there, with the closest one, we come to the blackboard. Show me something. Before then, I'll show you what an accurate and precise set of measures is. Let's say you have an axis here, this is the true measure, and these are units, whatever these units, congruent with this measure. If this is meters, you get it in meters. All right, so let's say we measure this property of the object we are trying to assess, and we get a number of measures. Most of the measures will fall close to the true measure. Some of them will be more distant. Very few of them will be even more distant. At some point we won't find any more measures. All right, so if we make enough measurements, we'll have a distribution which is Gaussian, which means the error in each measure is completely random. And so the distribution follows the curve of Gauss. All right, so some weather now will show me, will show us, on the same axis, what he thinks that accurate but not precise measures would look like. Accurate but not precise. You can look there and please come to the blackboard. Enjoy.
Accurate means that the average it's around here.
Right.
Like if it's not precise, I imagine this to be shifted. If it's not accurate, I imagine this to be more flat, less high, less high number.
I think you are correct, except that you exchanged accuracy with precision. It's the opposite of what you said, but I think you had the right answer in mind. You just said a different thing. So please draw.
So it's like shifted.
Okay, compare your curve with this. What do you say of those measures? Are they accurate?
No.
They are not. All right.
Okay. Yeah.
Are they precise?
Yeah, because the error is in a Gaussian.
No, that's not enough.
No.
That only tells you if the distribution of the measures is Gaussian. That means the error is random, is casual. But you have to look at how narrow the curve is.
It's the okay.
Right. So this curve is narrower than this.
Yeah.
So in fact, these measurements
Are more precise.
exactly,
Okay.
exactly.
Okay.
Right?
Okay.
Do you agree, Valeria? Go ahead.
The white give
Width?
the
Yes.
width give the precision, and average, like where is
Yes.
this point,
Mhm.
the distance from the average to the true
From the true
value
value?
is the accuracy.
Accuracy, exactly.
Okay.
All right.
Thank
Good. So now Valeria will show the opposite. Measurements that are accurate but not precise.
Okay.
Yeah, more or less. You understand? Okay?
If this is the true value, it would have to be like,
Exactly. So the average is correct, let's say. It's very close to the true value. But the dispersion of the
so
measures
it's
is wide.
not precise.
And finally, Karim? Karim or Karim?
Karim.
Karim will come here and show measures that are neither precise nor accurate.
Not precise and not accurate.
Excellent. Yes.
Yeah.
So we have a wide curve,
Yeah.
low precision, and a large distance from the true value, so low accuracy. Great. Thanks. You got the point. You all got the point. Now, of course, this shows you that the two, the two measures, the two properties are entirely separated from each other, and they can be present in either combination in a set of measurements. Now, let's look at the meaning of accuracy and precision. Of course, accuracy, an instrument, a method, or a set of measures, is accurate when it is close to the true value. We have said that. Precision is, instead, we have precision when the measures oscillate as little as possible. There is little variation between them. Now, in what situation would, let's see, in what situation would you have high accuracy but low precision?
When you find the right result, but
But
you
what
always
causes
have
this
you
low
have
precision?
like a wide error range
Exactly.
of error. So it's like most of the measures that you get are very similar to the true value, but you will still have a lot of values quite far from it.
Correct. Is the error you're talking about random or systematic? This requires a little thinking on your side.
It's human mistake, I guess.
I'm
So
not
it's
asking for an
random.
explanation
Okay.
yet. But
Okay.
I'm just asking, is it random or
Yes.
systematic?
I think it's random because if it were systematic, we would still have
What would you have?
a narrower. No, I don't know. It's.
If it were systematic, would you have high precision, do you think?
Yes,
Yes, you would.
but we would have less accuracy.
Exactly. This is a systematic error.
Okay.
You always have the same error, like a couple of
Yes. Okay.
points here, or units. But the measures are all close to one another. The opposite. Let's ask someone. If we have low precision but high accuracy, what do we conclude?
Low precision but high accuracy.
Something like this.
That our instrument is not that good. Like we're trying to measure a centimeter with a meter. So
Possibly, yes. So, for whatever reason, again, I'm not asking for specific reasons yet, but then we'll make an exercise about that. For whatever reason, the instrument is not in the same nominal conditions while measuring the same thing.
yeah,
Is not yielding the same measure, it's very similar measures all the time. So, for some reason, the instrument oscillates, the response oscillates. So, that could be due to what? When is your instrument oscillating? You gave a good explanation. So you're using an instrument which is not adequate for what you're trying to measure. So the error is large.
yeah,
And yet, overall, because it's a random error, you have an accurate set of measures, provided you measure the thing enough times that you
the average is
have a good average. Right.
yeah.
Right. And now, Karim, the opposite. when would you have this situation? High precision, low accuracy. We have said it in part, it's a systematic error. What kind of systematic error? There might be many, just try to think one or one.
Maybe a wrong protocol.
A wrong product, yes,
Maybe.
very possible. So, say you're making a colorimetric measurement. So, if you don't know that one substance, one substance in the thing you're measuring, in the solution you're measuring, influences the color,
I always have.
you always
wrong,
have the same influence, which shifts the average, even though the measurements, the measures are close to one another, and so they're precise, right? So, the distribution of measures tells you something about the error, if there is any. And now an even more difficult question. We have said this is precise, and we have said this is not. How close need to be the measures with one another for us to conclude that they are precise? How narrow should this curve be in order for us to conclude that the measures are precise? This is a difficult one, so think about it.
We have a standard to
We do
compare.
always have a standard. This is the standard.
Okay.
I think I remember in the chemical analytica exam,
Analytical chemistry,
it was
yes.
like the normal distribution needs to be like the how do you say larghezza?
Width.
Needs to be like 1/2 of the height.
Right, to be a true normal distribution, the curve has certain proportions, correct.
Okay.
But that's, you know, a mathematical requirement.
Okay.
What about a physical requirement? Which is, in fact, what I'm asking about.
Okay.
When are you satisfied by your measures? In other words, how close should the shot be to the center of the target for you to conclude that you are a good shooter?
Doesn't it depends on what you need your result for?
Exactly, exactly. There is no rule. It depends on what you need to do with your measures. If you need very, very accurate measures, you might require an error that is 0.001% of the average. But if you are weighing, say, flour for making a cake, you don't care if you have 10 or 10 grams of flour more or less. The cake will be the same, right? So it really depends. And depends on you, on your judgment. Depends on what you need to do with the measures you are acquiring. Right? So, very good. Next, robustness. Robustness is the ability, this is a characteristic of the method and or of the instrument. It's not a characteristic of the measure itself. So the method and or the instrument are robust when they are resistant to external influences capable of affecting the measure. So to give you an example, a typical analytical scale that you will find in the lab is not robust to vibrations. If you put it on a vibrating table, say because you have a centrifuge next to it, its zero will oscillate, so you will probably get a noisy readout, and your measure will be imprecise because of this oscillation given by the vibrations. So the typical laboratory scale is not robust to vibration. It is instead fairly resistant, fairly robust to temperature, provided that you zero the balance, the scale, at the same temperature as the temperature when you make the measurement. It would be okay between, I don't know, 10 and 40 degrees centigrade. It won't make a difference provided you zero properly the scale. Okay, and this is not a fixed property of the method or of the instrument. You can modify the method or the instrument to make it robust to whatever external influence you might have in your conditions. For example, say you have an instrument that must work not in the laboratory, in a relatively protected environment, but rather in the field, outside, wherever you are. So, in this case, you need an instrument that is robust to, say, the wind, because there might be windy situations outside the laboratory. It must be robust to some humidity. It may rain a bit. Things you don't care about in the laboratory. So perhaps you'll make the instrument heavier in order to resist the wind. You'll make it impermeable to resist the rain, etc. That tells us that robustness is not an absolute property of a method or of an instrument, but it's something that, first of all, relates to a specific external source of interference. There is no absolutely robust instrument. There are only instruments that are robust to certain influences and are not robust to others. And we can modify them in order to be robust to the conditions we are likely to encounter. For example, we are talking about the laboratory scale on a vibrating plane. Okay, so we put the scale on an anti-vibration plane that absorbs vibration. Now the instrument is robust to vibrations. Say you have unsuppressible vibrations. You can move away the centrifuge, so you don't need an anti-vibration table. But if, say, you have heavy traffic just outside the laboratory, the floor will vibrate whatever you do. So you have to modify the instrument or its setting in order to make the instrument robust to vibrations, and so on. So now a question for you. Suppose this is a measuring meter, and suppose I want to measure this height of what? Of this bar here. Okay. So I measure it, and I find that the measure is so much. 1 meter 70 centimeters. Okay. So suppose now that Valeria comes here, she does the same operation, and she finds 1 meter 71 centimeters. Now Valeria is just as accurate, it's just as good as I am at taking measurements. And yet her result, or a set of results, many results, have a different average as mine. Why might it be? Look at the act of measuring. This is the act of measuring.
Because I'm smaller.
Because you are shorter, you will do something like this. And so you make a mistake, a parallax mistake. So this method is not robust to the height of the measurer. And so, in order to make the method robust, we should include in the method that the eyes of the measure should be at the same height as the thing to be measured. So you jump on something and then you do the measurement properly. Good. Next, limit of detection. Oh, let's stop here. Let's stop here and talk about the best practice. First of all, even before then, the official description of robustness. This table is not perfect, but it gives you a good idea of what we're talking about. It's the lack of sensitivity to unintended changes, what we call external influences, in experimental reagents and protocols. So here it's talking only about reagents and protocols. They're implicitly thinking of a laboratory situation. But reagents and protocols are external to the instrument. So again, they are external influences. If your reagents are not good, the instrument won't work properly. Same as the protocol. If the protocol doesn't include the height of the eyes of the observer, then the method is not good. It's not robust. Okay, and let's now look at the best practice that this table suggests you to follow in order to attain the best possible accuracy, precision, etc. So, when you're looking for, when you are testing the accuracy of your measures or your measurement instruments, test your experimental observation using orthogonal analytical methods. That is, if possible, measure the same characteristic, the same variable, with orthogonal independent methods. If you reach the same conclusion, both methods are accurate. If you don't reach the same measure, with error, of course, it means at least one of the instruments is not accurate, or possibly both. Of course, you have to use well-defined reference materials to check instrument response and method validity. If you are not sure enough of your true value, there is no measuring, there is no determining accuracy. All right. As far as precision, replicate the measurement in your own lab, and this is what is most commonly done. That is, you want to make sure that your measures are really precise. You measure the same thing over and over again, and then you average them out, etc. Perhaps with different personnel, because there might be a systematic error due to, for example, the height of the observer, as we said. Or because one person is more skilled than another and makes better measurements. And then, perhaps, if you need to have a kind of measurement that is universally acceptable, then you have to run inter-laboratory comparison studies, which means you send exactly the same material to different laboratories and see what's the difference between the measures in the various laboratories. This is what the so-called reference laboratories do. When you need to be accurate about something, you set reference laboratories in each country, for example, and they are the reference, meaning that they've gone through this, they go actually periodically through inter-laboratory comparison studies and make sure that their measurements are as accurate as needed, or as precise as needed, or whatever is needed compared with the others. All right. Robustness. What's the best practice? What's the advice to the practitioner? Test different sources of reagents, fixation conditions. They're thinking about probably histological practice. Incubation times, if you're doing chemical reactions or biochemical reactions. Cell densities, if you're dealing with cells, and analysis software. So test whatever you think might be a source of instability in your measures. And we go through a practical test of what we are discussing here. Next characteristic, next measurement element, it is called in the table. Limit of detection. It should be clear enough. How low can we go with an instrument? So, limit of detection is defined as given the noise in the measurement, there might be a noise, there often is a noise in your measurement, the level below which the response is not meaningful. Which means, if, suppose this is the line of zero, right? Suppose your instrument is oscillating this much. A measure this high won't be significant because it's within the oscillation of the instrument. You don't really know whether this is pure noise or it contains a little bit of something you actually measure plus the noise in that precise instant. So you really don't know what to make of your measure. And there is no averaging in that case. It's really difficult to get a measure. So that's the limit of detection. Below a certain limit, you don't detect anything. Usually this limit, the limit can be either calculated statistically, meaning it should be at least, say, 2 standard deviations above the noise. Or you could say that it should be at least 3-fold as high as the noise. Whatever. You decide, again, in this case, what is your limit of detection depending on the need you have for your measures. I will add something else, but it won't be part of the exam, it's not on the book. There is also a limit of measurement. The limit of measurement, of measurability, is higher than the limit of detection. With an instrument I can detect that something, for example, say I'm using mass spectrometer. This is a good example. Say I'm using a mass spectrometer to determine whether a protein is or is not in my cell extract. Okay. So if there is too little of that protein, I won't see it, period. There might be just enough that I can detect a signal, but it's really at background level. So if I do it several times, I can convince myself that I can see that protein, because every time I see a peak just where it should be if the proteins were there. And yet that peak does not allow me to measure how much protein is there, because it's within the range of the noise. So again, I don't know what is the noise in the precise instant in which I see the protein. Whether it adds to the value of the protein, or it's actually negative and subtracts from the value, the true value of the protein. So the limit of measurement is higher than the limit of detection, because it needs to be high enough that the noise influences it sufficiently little. What is sufficiently little? You decide. Okay, how do we determine the limit of detection? Let's see what the best practice here. Use appropriate positive and negative controls, which tells you, by the way, that you can use positive and negative controls even in measurements, not only in experiments and observations. To determine background signal, a negative control that gives a whatever signal, of course, is yielding background signal. Doesn't come from the sample you are measuring. And use dispersion in replicate measurements to determine measurement uncertainty. which is what we have done here at the blackboard. Good. Next, response function. This is a bit complex. Dependence of signal on systematic change in experimental condition. Let's see a simple case. Suppose you are measuring the volume of a solution using a cylinder. Oftentimes, an instrument doesn't measure really directly the variable, the characteristic you're interested in. It measures a proxy, a derivative of that characteristic. For example, using a cylinder, you're actually not measuring a volume. You are measuring the distance between the top level, so-called menisc, of the fluid and the bottom of the cylinder. But you know that because this is a cylinder and the section of the cylinder is always the same, if the distance from the top of the fluid from the bottom doubles, then also the volume doubles, right? If it triples, then also the volume is tripled, etc. So you can mark the cylinder directly in milliliters based on the distance from the bottom that the fluid reaches. Clear? Okay. So in this case, the response function is simple. It's linear. It's a simple line. In other words, there is a direct correlation between what we measure, the distance, and what we want to measure, the volume. But you might have often, you have in your kitchen, volume measures that are not cylinders. In the kitchen, you don't use cylinders. You use stuff like this, for example, or anything, with a more complex shape than a cylinder. So, say it is a cone like this, then the curve is different. You are still measuring the distance between the top of the fluid and the bottom of the cone in this case. But you see, the first 10 milliliters really make a long distance from the bottom. The next one don't go as high because they spread on a larger width. The third one's even less, and so on. So the response curve is more complex, like this. This is actually a calculation I made with Excel, so it took an hour to make up this pieces, but it was worth the time. So you should not expect that your instrument always yields a linear relation with your variable of truth. For example, here is a more complex example. Let me stand for a little quiet. Per favore, potete parlare un po' di
Lezioni riguardanti.
So, what is this picture about? This is about real-time PCR. Do you know the principle of real-time PCR? More or less, approximately. Approximately, okay. Let's summarize it. Okay, in real-time PCR, as in regular PCR, you amplify a given amplitude. It is a short sequence of nucleotides. But in real-time PCR, the amplified material, in the simplest form, reacts with the fluorochrome, which is freely floating in the solution which PCR happens. So the more product, the more newly amplified DNA you have, the higher the fluorescence produced. And so you measure the fluorescence of the solution at every cycle. First cycle, second cycle, third cycle, 20th cycle, 25th cycle, etc. And there is a way to relate, which we won't discuss here, to relate that fluorescence with we have a noisy environment today. And there is a way to relate the intensity of fluorescence at a certain predetermined point with the starting amount of material. So you can retrospectively tell how much DNA, how much of that sequence you are interested in was present in the starting material by the time it takes to reach a certain amount of fluorescence by progressively amplifying. Right? So let's look at the response function of a real-time PCR. First of all, in the first n cycles, it depends on the concentration of the starting material. In the first n cycles, you don't see anything. The curve is flat, it's perfectly flat. Then at some point, it starts to go up, and a little later becomes essentially linear. From here to here, we have essentially a line. So in this range, within this range, the curve is in fact a linear correlation between the initial amount of material and the number of cycles necessary to amplify it to a certain point. Then you get a plateau phase. That is, even if you run the PCR further, the fluorescence increases little or no more. How do we explain a curve like this? This is a sigmoid curve, looks like an S, and it's fairly common in instruments. How do we explain it? Well, the starting phase is clear enough. There is simply not enough fluorescence to be detected by the detectors in the instrument. At some point, there will be enough fluorescence to detect. So we will be, in fact, at the limit of detection of the detector in the instrument. And from that point, indeed, because you double the amount of DNA replicated in the PCR at each cycle, from that point, for a while, at each cycle, in fact, the amount of fluorescence will double as well. So the relationship, the linear relationship is here. The amount of DNA in the amplicon doubles, fluorescence doubles. The amount of DNA triples, fluorescence triples, and so on. At some point, though, you either run out of reagents, say nucleotides, or polymerase, or something is exhausted in the reaction, so you no longer amplify anything, even if you run the PCR further, or the detector itself is not capable of detecting more than so much fluorescence. It's the detector itself that plateaus. And so this explains the shape of this curve. You have to know the response function of your instrument in order to correctly interpret its results. Because if you don't know what to expect, you might say, well, I've run the PCR up to 15 cycles, I'm detecting nothing, there is nothing. But you know, because I've seen this curve, that you have to wait to draw your conclusions, you have to wait long enough for the curve to go up. If it never goes up, then you have nothing in the beginning. But you have to wait long enough, not just run a few cycles and conclude that there was nothing. Similarly, in this phase here, you should not detect that there is this much material in the starting solution. There might be much more, but you aren't able to read it. So make sure that you are within, in this case, and not necessarily in other cases, within the linear range of your instrument. That is, you are in the range in which the relationship between what you read, fluorescence in this case, directly relates to the property you're interested in, amount of material of DNA in the starting solution. Okay, so this is very important. It is very important because you can make mistakes. We'll talk about that also later. We'll try to see a few applications of these concepts to practical situations. Finally, specificity. What? Yes. What do you do to understand your response function? Well, clearly enough. Systematically test concentration or activity with reference samples. So you have starting materials with known amounts of the sequence you want to amplify. You know how much you have in the beginning, and you record whatever readout you get from your PCR. If you have 1 nanogram of starting material, let's say, you'll find that perhaps you need 10 cycles to begin to see fluorescence increase. If you have 1 picogram, you'll probably need 15 to 20 cycles to see something. If you have less, even more cycles and so on. And determine the range in which the assay is sensitive. The assay is sensitive in this range, rather on the y-axis. Lower than that, it is not sensitive because you do not detect any fluorescence. Higher than that, it is not sensitive to any variations. You have a maximum readout, and then beyond that, you are unable to read more. Now, just a detail, and then we'll go on. You see that the initial phase here is not 0. It's 1 on a Hardy-Fry scale. Why is it 1 and not 0?
Because I have the sample.
Because you have the sample. For example, yes, it is possible that the fluorochrome is reacting with the sample itself. Yes. Or it is possible that you have so-called electronic noise. That the detector itself is reading something that is not there, so it is not 0 property. And there are other possibilities, but basically this is background. Your background in the PCR is not 0. All right. Next, specificity. The analytical result is not confounded by sample composition or physical characteristics. This is very interesting because, again, you have to be careful evaluating the circumstances in which you do your measurements. And in one of the next lessons, we'll see a rather complex example of this. But to understand, I think, intuitively what specificity is. If I want to measure, for example, a certain amount of DNA, I don't want my reaction, my measurement, to be interfered with by, say, external material. Say, for example, other sequences that are amplified spuriously, non-specifically. Because, for example, my oligos have not been designed properly and they align elsewhere. Or, you might have, this is even more important, you might have in your PCR substances that interfere with the readout. For example, autofluorescent substances that have their own fluorescence, and so they artificially raise the readout of the PCR. Or much more important, you might have inhibitors of the PCR reaction, so that the reaction is much less efficient. And if you compare it and you don't know about that, for example, because you extracted your starting material, your DNA, from a dirty sample. Typical dirty sample in the laboratory is mouse tails. In order to determine the genotype of a laboratory mouse, you cut the tip of the tail from the mouse. The mouse doesn't feel anything, it doesn't even squint. But you take the DNA from this tip straight away with simple protocols, but nonetheless sometimes you dirty DNA. Dirty DNA, dirty meaning contaminated by substances that inhibit the reaction, the PCR reaction. So in these cases, you might fail to detect anything in your PCR. Or, and this is basically a qualitative kind of determination. You only want to know whether the genotype of the mouse is, say, heterozygous or homozygous. So either you have two bands or one band. You don't care how much. But if you are trying to determine how many copies of a certain gene are in the DNA of the mouse, for example, because you have gene duplication, say in a tumor that developed in that mouse, then you need to have very clean DNA, because if, say, the DNA contains inhibitors of the PCR reaction, not inhibitors that block the reaction altogether, but simply reduce its efficiency, you get a curve that goes like this. It rises very slowly, and you are brought to erroneously conclude that there are very few copies of the DNA of the gene, or whatever you're looking for, in the starting material, but there are many more, but you don't know that the efficiency of your PCR has been lowered by contamination. So, you have to make sure, in general, that your results are not affected by what is called the matrix effect, which is not a reference to the movie. It's, in fact, the matrix is whatever is not your analyte. So, for example, in a cell extract, in which you want to determine the amount of a certain protein, everything else is the matrix. So there is much more matrix than your analyte. And you have to make sure that either the matrix does not interfere with your measurement, because your measurement is robust to the kind of matrix you have at hand, or that you can measure the influence of the matrix and include it in the calculations that bring you to the measure. So, for example, you might have a matrix that, as I mentioned, say in a fluorimetric assay, in which you use a reagent that reacts with, again, it could be DNA. In a fluorimetric assay, you add the reagent in a sample, and then you measure with the fluorimeter, you measure the amount of fluorescence produced by the sample. Now, some matrices might absorb fluorescence, and so give a lower readout than would be expected. You have to know that, because again, otherwise you draw wrong conclusions. On the opposite, and this is really specificity, you might have substances that do react with the fluorochrome, like DNA does. And in that case, you overestimate the amount of DNA. That's no good either. In that case, you lack specificity. Your reagent, which is supposed to be specific for DNA, in fact reacts with something else and reads other things. Now, this is very common in in the laboratory. It is extremely common. It's also very common in clinical laboratory analysis. That's why, for example, they tell you that you should be that you should not eat before giving blood for laboratory analysis, because what you have in your blood interferes with the analysis, interferes with the result. And so, what's the advice about the best practice? When testing samples from different sources, ensure that apparent response differences are not due to sample matrix differences by using spiking controls. What are spiking controls? Spike is a jargon. It originally means adding alcohol to a beverage. In the laboratory, it means in my unknown or undefined sample, in which I don't know the matrix effect, whether there is any matrix effect, and if there is one, how much it affects my measure, I add a known amount of the analyte. Because I know how much analyte I've added, then I know exactly what to expect, assuming there is no matrix effect. If I don't get what I expected, then I know that that is due to the matrix effect, and I can quantify the matrix effect. And then I can bear that I can put it into the calculations that bring me to a result. That is, for example, I know my matrix absorbs or reduces by 50% my measurement. Okay, I'll just double whatever measure I get, because I know it's halved by the matrix. But I have to have determined it in advance.
So it's like a control to...
It is like a positive control, yes. All right. These are the characteristics I wanted to talk about. And this column here, which sometimes I don't read it in the lesson, is instead very important because if you think a little about it, it gives you a lot of suggestion as to how to proceed in practical circumstances. And we'll see in the next lesson that it is extremely important to know the range in which your measurements are valid, what affects them, how to take such influences into account or eliminate those influences by making your method more robust, and so on. If you don't know these things, if you use a measurement instrument as a black box, then put a sample here and get an answer there, then you run very serious risks of going to the wrong conclusions. Again, we'll see real examples of that. Finally, technical variability. We have already discussed a bit about that when we talked about controls, negative controls in particular. So, perhaps Karim can remind us what technical variability is, as opposed to biological variability, which is what we certainly encounter in the biological lab.
I don't remember, to be honest, the last lesson.
It's okay. Does anybody remember, or is anybody willing to provide a guess?
It's not like every instrument is able to measure the same measures, I guess. Like if we have a spectrophotometer, not all of them maybe are able to analyze the same wavelength.
Okay, yes, but that's not technical variability.
Okay.
Technical variability exists within a single method or a single instrument. And I was opposing it to biological variability. So if you measure a number of biological samples, they won't be identical to one another. So certainly you will have different measures. And that's biological variability. What is technical variability?
If you do more measure, it's not sure that are all the same, even
Or
with
measure
the-
so what?
of a nanolite, no,
Yeah.
of a nanolite. Yeah.
An analyte, one analyte. That's what you're saying?
Yeah.
Yeah, that's correct.
Okay.
That's technical viability. So, if you measure several times the same analyte, not different ones, there is no biological variability because the analyte is the same. Then whatever variability you observe must be technical, must be due to your instrument or your method. Okay. So, we already reached the conclusion here, but let's go through the slide
Is
and
that
see
like
what...
the error of the instrument?
Yes, it's
Ah.
the error of the instrument, the technical error of the instrument. Or you can actually talk about the error of the instrument, because there is no biological side on in the instrument. Okay, here is the example I wanted to show you, but we have already come to the conclusion, I'm sure that you give me the right answer at the end of this little test. Okay, let's say we are analyzing two theoretical identical biological samples. Say these are two cell extracts coming from two identical cultures that we extracted separately, right? So in principle they should be identical. We want to know what proteins they contain. In order to do that we use mass spectrometry. Mass spectrometry is extremely powerful but has some limitations. In very complex measures, not always it is capable to see everything that's in your sample. Sometimes it takes out let's say, gives you a partial response simply because there are too many proteins at the same time and the instrument can't see them all. So let's say we measure these two samples and we see that most identified proteins are in common. So we identify 350 proteins both in the first and in the second sample. But we have 41 extra proteins in the green sample, not found in the red sample, and 37 in the red sample, not found in the green sample. So, we wonder how much of this variability how much of this variability is biological variability and how much is technical? That's the question for you. What should you do to determine what is the technical variability and what is the biological variability?
A technical control and a biological control.
Half correct. The first part is correct.
Okay.
The second part is impossible.
Okay.
I'll show you why. Okay, the technical control would be what?
Unknown quantity of protein, unknown mixture of protein.
It might be, or doesn't even need to be known.
Aspected the amount of protein.
Not necessary. It could be a completely undescribed, unknown sample.
We just
You have
measure-
already said how to do a technical
We
control.
just measure it like 10 times.
Exactly, you said that. Fortunately, Valeria remembered. So instead of measuring two samples, remember this very simple equation. Technical variability is equal to total variation minus technical variation. Biological variation. It's a mistake. Okay, remember this is biological.
Okay.
Okay, we can measure biological variation, or it's very, very difficult. How do we know what is the difference, say, in gene expression in these two apparently identical cultures? What affects them, whether it is time or conditions? It's extremely difficult to assess biological variation, but it's easy to assess technical variation. variation. We simply take a single sample instead of one. We measure it twice, or 10 times, or 20 times, depending on how accurate we want to be. And then we find, for example, that the technical variability is much smaller than the total variability here. 47 instead of 41, 12 instead of 37. So this is technical variation. The rest must be biological.
Oh, okay.
All right, so again, this is important in practice because when you are confronted with variation, you need to understand what is due to your instrument. If your instrument is not precise enough, you may want to change it. You may want to have it repaired. You may want to use a different method, whatever. Because technical variability, in principle, can be suppressed. Not entirely, but in large part. Biological variability is exactly what you're measuring, so there's nothing that you can do about it. Good. So this concludes this lesson. But because we have time, we'll go to the next one, which is very much related to this. Okay. In this lesson, after talking about measurements and measure, we'll talk about the all-important measurement instruments. And so we'll discuss how to validate a measurement instrument. So I'll present you with a realistic scenario, and you will tell me what I should do, or what you would do in that situation. So, say a representative of a instrument factory comes to your lab, talks to you, and suggests that you should buy an automatic cell counter. What is an automatic cell counter? It's an instrument that saves a lot of time in laboratories that make many cell counts. Because doing them by hand it's tedious, it takes time, and so people may want to have it automated. And so the representative really talks enthusiastically about the properties of the instrument. It's magic, it makes your life easy, and you should get it, absolutely, and convinces you that it's a great instrument to have. So you go to the lab chief and say, you know, we really should buy this instrument. And the lab chief says, well, maybe we should, but you have to convince me that it is true, that we should buy it. So I want to know that this instrument is really accurate. I want to know that it is precise. So why is the laboratory chief asking for that? Because he wants to know that the measures he gets are at least as accurate as the ones you usually get with the ordinary procedures. Otherwise the instrument wouldn't match the ordinary, the standards the laboratory is already following. He wants to know that the measures are precise, because if they are not, you have to measure the same sample many times in order to have an average, like here, here, that that matches the actual number of cells. All right, so how do you determine whether this instrument is accurate and precise or not? What would you do?
With an orthogonal method of analysis.
Yes, such as? Have you ever counted cells?
In a solution.
Yeah, in suspension.
Can we use like spectrophotometer?
You can.
We obtain
You've done it with bacteria, right?
Yes,
Right.
optical density of the. So, how
Yeah.
would you say? Not solution.
Suspension.
Suspension. Yes.
Yeah, that's a possibility, absolutely. So if you're dealing with bacteria, you can do that. Keeping in mind, though, that absorption is a rather indirect measurement in terms of as it relates to the number of cells. If they are bigger, for example, they will have the same absorption, but there will be fewer. Of the opposite, if they are smaller, and if they are contaminant, blah 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 blah. So you should be as close as possible to the actual quantity you want to measure, rather than using indirect methods. Indirect methods, in general, are more prone to error. But it's a good possibility, yes. So it was a good answer. And in any case, here we are not dealing with bacteria, but with eukaryotic cells. So we need a different method.
Maybe try the instrument with something that I know already the true value,
Of course.
look.
That's the only way to determine accuracy. You have to know a true value. Now, the problem is, how do you determine a true value of a cell suspension? How many cells are there, say, cells per milliliter in this suspension?
Dilution and
Dilution, and how do you count them?
on a plate.
On a plate,
With
yes,
directive method,
it would take a week to get
Okay.
an answer. It would be tedious because you would have to repeat it several times to assess accuracy and precision. So the faster way. Also, colony formation, which is what you're thinking of,
Yeah.
is affected by the efficiency with which the cells grow in those conditions. Not all of the cells plated generally grow. So you underestimate systematically the number of cells if you just count colonies that raise from them. You've never heard of this very common method in the laboratory, the hemocytometer? No. Then I'll show you.
Flow cytometer.
Flow cytometer is
Yeah.
complex instrument. This is a very simple one. I'll show you how it works. It's been invented in the 19th century. So basically you have a glass plate the size of a microscopic slide, and you see here in section. So you have a plane here, which is where you put the sample. This plate here is at a distance with a cover slide, a thin slide that you put on top, in order to keep this volume constant. And these are just sideway for the overflow. If you overload the chamber, it will flow away from there. So, how do you load the chamber? So, you have this little instrument here. You put a cover slip onto it, here, and then with a micropipette, you slide your cell suspension underneath the cover slide. And you basically fill up this volume with your cell suspension. Okay? Now, let's enlarge this section here. This section is identical to this one, so you can do two measurements without washing the hemocytometer. This section here contains a grid like this, which is made by really make very thin incision in the glass. And this is a 1 millimeter square, further subdivided into smaller squares. And basically, you count the number of cells that are within this square, and you multiply it by a proper factor. How much? Here it is. I forgot to mention. The distance between the plate here and the cover slip of the glass is 0.1 millimeter. The size of the square you are counting cell in is 1 square millimeter. What's the volume of the suspension in this area?
0.1 millimeter square.
0.1 cubic millimeter. Yes, correct. In liters, how much is it? In microliters.
1 liter is 1 decimeter cubic. So, un litro è
Cubic
decimetro
decimeter.
cubo.
Yes,
Just-
yes. Right. Let's do it here. See, this is a rare instance in which a biology student remembers the equivalence that he studied in elementary school. Most of you don't remember how to do that. Okay, so we have 0.1 cubic millimeter. And so you say 1 liter is 1 cubic decimeter. Yes.
So we have 0.1 millimeter in decimeter is per 10 to the power of 5.
More than that. Much more.
Yeah, because
Because they are cubic measures.
yeah, but they are both cubic, so it doesn't work.
They are both cubic, correct? But going from millimeters to decimeter in linear measures is only what? 100-fold, right?
Yeah.
10 millimeters make a centimeter, 10 centimeters make a decimeter, and that's it. So the distance is 100 between
Yeah.
these two. But in cubic,
Yeah, I
measures-
multiplied per 3, so to the 5, but it's not.
no?
It's elevated as 3.
Exactly.
Yeah.
Exactly. So it's
To the power of
3.
3 over 2, 6.
2
No.
to the power of 3 is
8.
8.
Yeah.
Okay. So 8-fold. Between millimeter and decimeter. But then this is 0.1 cubic millimeter.
So 9.
Yes, correct.
So it's 1 picoliter. I don't know if it's
1
pi-
microliter.
micro.
Microliter. Yes, correct. So, what you do basically is you count these cells, and then you, it's not here, you multiply it by that factor, and you come to the conclusion that say, let's see, we have 9, 6, 15, 20, 25 cells, 24 cells here, times 10 to the 9th, and you determine what's the number of cells in 1 milliliter of the original suspension. Alright, that's what one could call a gold standard. What is a gold standard? A gold standard is the best method and the best instrument that you have to determine the variable that you're interested in measuring. The hemocytometer arguably is the gold standard in counting cells because it's very simple, both in principle and in its actual structural form. And so it has the fewest chances of being affected by error. The principle is pretty obvious once you know it. So that's very reliable. So you'll never know exactly how many cells you have in your suspension. But the best measurement, in fact the best count, which is slightly different from measuring, the best count you can get is by using the hemocytometer. So that's your true measurement. Then, having a known sample with a true measurement, what would you do? To address the questions that your laboratory chief asked you.
Try the new instrument
Okay.
and see the difference between the value that the instrument gave and our standard.
Exactly.
And for the precision, I will do it many times
Exactly.
and see how much this.
Both here and with the automatic cytometer.
Okay,
Because
yeah.
you can compare them and see which one of them is more precise. Right? Good. Really good. Okay, let's talk about accuracy now. Let's say you determine the number of cells in the same suspension several times with the hemocytometer and with the automatic cell counter. And you get very similar measures, but not identical, of course. Say with the automatic cytometer you get an average of 67 cells per whatever, microliter or whatever it is. And with the manual hemocytometer you get an average of 68. Now, and you see, the distribution is pretty much the same. It's Gaussian in both cases. So you know that the error is only casual, it's random, it's not systematic. Or it might be, okay, let's say the distribution is certainly affected only by random error. The difference between the averages might be true or not. The question is this a significant difference or is it not? What are you going to tell your lab chief? Okay, it works just as fine as the hemocytometer. Or, well, it is not really accurate as the hemocytometer.
Yeah, it's significant difference. Because
Good question. Is it a significant difference? You can tell from this.
okay.
But that's the right question to answer. So there are statistical ways we'll talk about that.
Yeah, I thought about seeing the curve, like, for be significant, I imagine, like, the peak really more close than, like, a full standard deviation, like.
That's just a crude approximation.
Okay.
No. There are specific techniques, statistical techniques to determine whether this difference is significant or not. You have to know, though, each single measure and the distribution of them. You have to have a full table of measures, and then you can calculate the statistics. But we won't do it here. Let's say that your statistical evaluation says that these two averages are in fact the same, that the difference is just causal. If you did the experiment twice, maybe they would exchange, there would be a similar difference, but it is really not significant. Good. Then you conclude that the two instruments are just as accurate. They're both, they have the same accuracy and the same precision, right? Same accuracy because the difference in the average is insignificant, and same precision because the shape of the curves is the same. So, good, then you go back to the lab chief and say, okay, we can buy it, I have the proof. Look here, I have the results. And the lab chief says, well, yes, good, but I'm still not convinced. Because you have to show me that this instrument is as robust as the hemocytometer in terms of being able to distinguish live from dead cells. Usually when you count cells, you are not interested in dead cells. You don't count them at all. In the hemocytometers, you see them, and you can stain even them. Stain only dead cells, not live cells, because they can't penetrate the membrane of live cells. But in dead cells, the membrane is permeable, and so they become stained. So it's easy to distinguish that from live cells. The automatic hemocytometer doesn't use that technique. It uses an indirect way to distinguish live from dead cells. It looks for cells that are smaller, less uniform, etc. It has a number of algorithms to distinguish them. And so, the new question your lab chief asks is the automatic hemocytometer as good as we are at the microscope in distinguishing live from dead cells? And then, how do you address that question? Well, how does Valeria address that question? You have been drilled enough today. The approach is similar.
We measure a sample full of dead cells, I don't know.
With a known number of dead cells, yeah.
And we do the measure with both instruments and see if our eye is better than the instrument.
So remember that the hemocytometer is our gold standard. We believe that until now, at least, this is the best instrument we can use, not only to count cells, but also to distinguish dead from live cells. And so we determine whether we get the same results with two instruments. All right. So if we get the same results, again, Samuele goes back to the lab chief, or you go instead of him because he's tired of all these experiments, and you both argue that the instrument is okay and we should buy it because it would save us a lot of time. On the other hand, if you get different results, say, for example, to simplify, say you get these two averages and your statistics show that the difference is significant. And in fact, the instruments are giving you different measures. What is the best one? What is the measure, the average closest to the actual, to the true value?
We believe our standard.
I repeat the maturation.
Yeah, and because they are statistically significant, you'll probably get the same difference, more or less, within the measure. So the question will stay the same. The answer is there is no way. Or there is no no unmistakable way to determine which one is the best. Perhaps, even if this difference is real, perhaps the automatic hemocytometer, the automatic cell counter is better than the hemocytometer. But only time will tell us. In fact, what decides what is the gold standard is common practice, is the scientific community that uses those instruments or those methods. They decide that at some point, after doing many experiments, running many different tests, that one method is the best, and that becomes the gold standard. But that's not forever. Somebody might build a better instrument, invent a better instrument, and so that becomes the new gold standard. So the situation here might be similar to this, but we have already discussed these possibilities. So it is what we drew at the blackboard before, so we won't go into this. So I I'd like to conclude this first part of this lesson by discussing a very significant and I think a very telling case that came out in the literature years ago. This is a small article published in Science in 2006. Okay. And describes a real situation in which this guy, Geoffrey Chang, who was a well-known structural biologist in a top lab in the United States, raised to a star fame as a structural biologist because he was very young, very capable, and published very bright structure in a short time. So he was really a rising star in the field. He was very good, but every time he published a new structure, a new protein structure, people were surprised. People who were dealing with senior proteins said, "Ooh, we would not have expected this protein to have that structure. We thought there was something different." Well, that might happen. So proteins that have similar primary structure might have different tertiary structures. But then this happened over and over again. Every time he published a new sequence, it was different from what people expected. And so that became a little suspicious until one of the people, of the scientists, surprised by one result published by Chang, looked harder at the data and saw what the problem was. Chang was publishing mirror images of the structure. And so he alerted Chang and said, there is a problem with your structures. And he went back to his calculations and basically found that a piece of software that he had found in the laboratory that had been created there, not bought, not available to anybody else, exchanged one column with another. And so created mirror results that eventually produce mirror images of the structure. And so he had to retract 5 papers. Now, this was an honest mistake. He didn't do it on purpose. He wasn't trying to cheat people. So it happens. It shouldn't happen, but sometimes it happens. Nobody's really condemned for that. But really, it was a strike to his career because he was highly reputed, and his reputation really fell because of this. So, what was Chang's mistake? What did he do wrong? In good faith. What did he do wrong? Karim will tell us.
The robustness of
It's not really robustness, but it's sort of. Perhaps he should have gone through the whole program to see whether there were any errors. No. Perhaps 1,000 lines of program and see whether there were any errors.
Maybe a systematic error. Yeah, this
It
is
was in the end a
yes.
systematic error,
Yeah.
but he didn't know. But are you? If I'm proposing that he should have gone through the program itself to determine whether any passage was correct, would you agree with me?
Yes, but I think he
Would be extremely time consuming and prone to error anyway. Have you ever tried to do that?
no.
No. Try. You won't see the mistake oftentimes. That's why people are debugging programs in a different way. They make them run, and whenever they encounter error, they stop there, and so the last line most likely is the source of the error. That's how you debug a program. Just reading it won't probably get you to the problem, if there is any. You don't even know in the beginning if there is a problem. So if you read it
Yeah.
and you don't find any problem, that's fine with you. But that's not a safe way to assess whether the program is reliable. What else could you do? Assume it is not a software, but whatever instrument. The software is itself an instrument.
Ask to another laboratory to repeat the experiments.
You might, but these are very heavy calculations, you know. I don't think people would do this favor to you. They would waste weeks to assess whether your instrument is okay. You do it. There must be a way. Otherwise, Cheng wouldn't have been even at fault. You just failed. Yeah. But he was at fault, although in good faith. He had not checked his instrument. He treated it as a black box. He said, the data go from here and they get the result there. I don't know and I don't want to know what happens. Of course, he was drawn to this mistake because he came to a laboratory where this piece of software was already in use. He inherited it. He did not contribute to its development. So he assumed it was okay. As you will in your next laboratory. They will tell you this instrument, use this instrument, and you get the results. You'll trust them. And 99% of the times it will be okay. But if you don't know exactly what you're doing, how the instrument works, you might get into this kind of trouble. A scientist's nightmare. So, what could have Chang done to test this software, to test this instrument? What you just suggested before.
To analyze protein, which structure we knew.
A known protein. See whether you got the proper structure. Exactly. See, just another thinking. You should never trust a piece of software. Just because it comes from Microsoft or from Apple, it's no guarantee. They actually do not guarantee that the software will work as advertised. They say you buy it at your own risk. You buy it, use it at your own risk. You'll find in the license. They cannot guarantee because there is a near infinite number of possibilities that something might go wrong. And in fact, you know that every once in a while something does go wrong. The program crashes, so something must have gone. And so he should have run a test. Yes, he should have run through the software, through the other softwares that he probably used, a known structure. That's the way you test your instrument. Now, that's an excellent way to test an instrument, that is to use a known standard to determine whether the instrument is working properly. The other excellent thing that you should always do is to know at least the principle on which the instrument works. Because the principles that are at the basis of the instrument. There are instruments so complex that only the physicists or the engineers that have invented them really know exactly what's going on in there. But you should know the principles. Because, for example, let me think of a fairly complex instrument. All right. Let's think of a nuclear magnetic resonance for taking images, say of humans or animals. In the laboratory we use instruments that are made for small animals rather than for humans. Instead of having a large bore in which you slide in, we have bores like this in which you can put a mouse or a rat. So, you have to know how those instruments work. They're absolutely too complex for you to understand in detail how they work. You should know some principles. You should know that they are not robust to movement. If the animal moves, you will get a lot of artifacts that might look normal. That is, you get abnormalities that you believe, which are not there because they're really artifacts due to movement. So you have to know that you have to deeply anesthetize the animal in order to make sure that it doesn't move. You have to know that these are magnetic, with a very strong magnetic field, instruments with a very strong magnetic field, which is affected by metals. So there should be no metal around the instrument, or by any chance, in the animal. Sometimes to distinguish an animal from another, a mouse from another, we mark them with little metal things in the ears. Well, that should not go into an NMR machine because you'll get a lot of artifacts, perhaps even dangerous, etc., etc. So you have to know at least the basic principles of your instrument in order to understand what are its limitations, what are the applications that might generate unreliable results. And if you really are not sure whether your situation is, whether your instrument is suitable or not in your specific situation, then you should run a test with a known standard. That's what you should do. And that's exactly what poor Cheng should have done. If he wasn't sure, if he had thought there was at least a chance that the software was wrong, he should have run a test. So remember, know your instruments. And that's it for today. Perfect.